On behalf of BioIT World's Global Web Symposia and our sponsor, Agena Bioscience, I'd like to welcome you all to today's program, Routine Pharmacogenetic Analysis Using a Robust Genotyping and Software Platform. I'm Allison Prophet, editor of BioIT World and the moderator for today's event. Joining us first is Dr. Stuart Scott, Assistant Professor of Genetics and Genetic Sciences at the Icahn School of Medicine at Mount Sinai. Welcome, Stuart.
Thanks, Allison. Thanks for the introduction, and I just wanted to thank Agena Bioscience for the opportunity to present to you guys today. So good morning, everybody. I'm going to talk today about pharmacogenetics and some of our work on implementation that we've been doing here at Mount Sinai. For an outline, it's going to start with a little bit of introduction and background on pharmacogenetics and the CYP450 system, and then talk a little bit about something that I'm interested in, the concept of pharmacoethnicity and how I think that's important when we consider clinical implementation of pharmacogenetics. And finally end off with some examples of how we are implementing pharmacogenetics here at Mount Sinai and some of the challenges we have faced and some of the issues that we have tried to address as we deploy clinical pharmacogenetic testing here in the preemptive model. So to start off with some background, I think everybody here has probably heard the terms precision and personalized medicine several times. It's kind of a buzz term. And there are several institutions that have institutes that are dedicated to this effort. Of course, there's the Personalized Medicine Coalition. We have an Institute for Personalized Medicine here at Mount Sinai, and many other institutions have their own initiatives ongoing. It's really been spearheaded as well by the recent NIH initiative to launch the Precision Medicine Initiative. So it's certainly something that I think everyone is very interested in these days, and it's something that there's a lot more funding that's being directed towards, and I think pharmacogenetics is going to play a very big role in that effort. One thing I'd like to start off with is looking back to 2009 when we talk about direct-to-consumer genetic testing and the personalized medicine companies that kind of sprung up at that time. And this opinion piece that Craig Venter published in Nature in 2009 where they compared the results from two different direct-to-consumer genetic testing companies on 13 diseases for 5 different individuals. And what they found was that for these disease risk prediction genetic testing companies, that there was very low concordance between them, and that was likely due to the discrepancies between the markers that they used. None of them used identical sets of markers. So they published these recommendations for direct-to-consumer testing companies at that time and for the personalized medicine field in general. And one of those recommendations was to include pharmacogenomic markers, because at that time they weren't really included in these testing panels. And they said that these were likely going to be the most actionable pharmacogenetic or pharmacomarkers out there that could probably be used in these precision medicine initiatives. So since then, a lot of these companies have started including pharmacogenetic markers, but I'd like to just mention that it's not just the direct-to-consumer testing companies, but more so the CLIA labs that should really be driving this area. As we've probably heard before, there's not just 23andMe, but several other companies have also gone direct-to-consumer to deploy pharmacogenetic testing. And some of them, unfortunately, have run into a little bit of trouble with how they are presenting these data and how they're offering this testing. Some of them also are being sent letters by the FDA with concerns over how they're going direct-to-consumer and not through a physician. So it's something that's an ongoing area of debate on how to deploy pharmacogenetic testing, whether it should be done direct-to-consumer or always through a physician. I'm personally of the mind that the physician should always be involved to ensure that the therapy on the other end is always being overseen by a professional and not the consumer themselves. That's a little bit beyond today's discussion. So pharmacogenetics and pharmacogenomics are often interchanged, but we can define pharmacogenetics as the study of drug response in relation to specific candidate genes versus pharmacogenomics, which is looking at the entire genome. And if we consider the number of publications on this area, the field started essentially in the 1950s, and where we see the rise in publications, particularly as the Genome Project was completed. And sequencing technology really prompted pharmacogenomics. But I also like to consider publications that were focused on clinical pharmacogenetics, because we see that lag between the publications on pharmacogenetics and then this lag where we talk about clinical implementation. And I think that really suggests there's this phase in between discovery and in clinical implementation, and a desire to kind of implement things that we identify. They have journals dedicated to them, like every other field, and it's something that we often see in the popular press now as well, too. But I think probably the biggest thing for the field was the 15 years of support from NIH that was directed towards the Pharmacogenomics Research Network that really launched and supported a lot of huge initiatives and research programs, as well as PharmGKB, and what we'll talk about a little bit later, the Clinical Pharmacogenetics Implementation Consortium, or CPIC. So why do we care at all about pharmacogenetics? And I think this is nicely shown here by the Personalized Medicine Coalition, looking at the percentage of patients for which a particular drug class is actually ineffective. Starting at the top, we can see antidepressants, asthma drugs, diabetes, arthritis drugs, Alzheimer's, all have a fairly high rate of inefficacy or a lack of efficacy for many patients. Of course, chemotherapy is a big one as well, too, but for the purpose of today, I'm going to avoid talking about any kind of chemotherapy agents just due to the complexity of that, the interaction between the constitutional genome versus the heterogeneity of the tumor genome, restricting my talk today to just constitutional pharmacogenetics. I think this has also been underscored, the importance of pharmacogenetics, when we look at a really nice review that Mary Relling published in Nature just last winter, looking at the number of FDA-approved medications that have actionable pharmacogenes listed in their label, and it's 7% out of all 1,200 pharmacogenetic genes, which equates to about 84, which is not a huge number, but when we translate that to the number of prescriptions in the United States on the right-hand side of these same medications that are FDA-approved that have actionable pharmacogenes associated with them, that number increases to 18%. And when we translate that to reconcile that with an actual number of prescriptions, it equals about 720 million prescriptions in the United States alone, which suggests that these are not just rare medications, that these are medications that are actually quite frequently prescribed. So the other thing I like to also include at the early stages of these talks is that we don't want to overhype pharmacogenetics. I think that's one area that often has left us getting into a little bit of hot water in certain situations, because we know that it's not just genetics that's contributing to variability in drug response, and there's a number of other factors, and we don't want to overhype our findings. And so I think when we put together genetics as well as age, race, ethnicity, other concomitant diseases and medications, this is all needs to be taken into consideration when we talk about drug response variability. And if we put these together, I think that's when we really get to this concept of precision medicine. So the enzymes, or the genes that we often study in the field of pharmacogenetics, are those involved in drug metabolism. And drug metabolism has artificially been broken out into phase 1 and phase 2 enzymes. And on the left showed the phase 1 enzymes, which are these kind of hydroxylation or reactions like that are often mediated by the C450 system. And then the phase 2 enzymes are usually involved in the larger kind of conjugation reactions like acetyltransferases and methyltransferases and things like that. But some of the first genes that were kind of included into clinical genetic testing panels were some of the P450s, most notably CYP2C9, 2C19, and 2D6. And the reason for that was because between the three of them, they were involved in about 40% of drug metabolism. So it was many years ago, many companies started to develop tests, genotyping tests for these three genes, some of which are FDA approved today. So the CYP450s, if you don't know about them already, are a very large, diverse superfamily of hemoproteins that catalyze the hydroxylation and other metabolic reactions. They are the major enzymes involved in drug metabolism and bioactivation, and thankfully, there is a resource for us to catalog the number of genetic variants in them because they're highly polymorphic genes. There's over 60 different P450 genes and pseudogenes, and they're very hard to study sometimes due to the sequence homology between them. The nomenclature is also quite confusing for some people sometimes because it uses the star allele system. So just to give you an example of what this represents, the CYP is the superfamily, the 2 would be the family, the C is the subfamily, and the 19 in this example is the isoform of that subfamily. And then the star 2 is actually representing the variant allele, which in this case is the 681G to A coding region transition variant that alters the splicing of CYP2C19 and encodes a loss of function enzyme. An important thing to note, though, is that these star alleles are actually haplotypes. So we often genotype the functional variants, but when we report out star alleles, we're actually inferring that there is a diplotype or two haplotypes there, and that the star 1 haplotype represents the normal functional copy of the enzyme, and all of the other star alleles usually represent the variant haplotypes, most often with some kind of reduced function or inactivity, but many of which we actually don't know their functional effects due to how rare they are. But as I mentioned, we can thankfully go to the Karolinska Institute's website to find out this kind of information. So once we have the genotype of the P450s, the next step is to infer our predicted phenotypes, our metabolic phenotypes. And there's kind of a standard system in place to do that, which is fairly straightforward for most of the genes, but not all of them. But the normal metabolizers, are previously referred to as extensive metabolizers, are individuals who have two normal copies of CYP2, or one of the P450s, in this case 2C9. Intermediate metabolizers are those who inherit one reduced function or loss of function allele, in this case the star 2 allele. And the poor metabolizers are those who are either homozygous or compound heterozygous for these loss of function alleles. But an important point I want to make here is that the difference between targeted genotyping and full gene sequencing. Because most laboratories will be doing targeted genotyping and restricting their analysis to those known variants where we have functional activity, which is very different from full gene sequencing because we're basically masking ourselves from the rest of the data or the rest of the sequence of that gene by doing targeted genotyping. So the star 1 haplotype is usually reported in the absence of the identification of any other variant allele, which is very important because if one laboratory is testing for only two variants like star 2 and star 3 and another is testing for a much larger panel, you could have some discrepancy between the results between these laboratories. We really don't know if somebody is a true star 1 unless we do the full gene sequencing, but the balance there is that you might come up with a lot of variants of unknown significance by doing full gene sequencing. So I'm of the belief that we should still be doing just targeted testing to those functional variants that we know the actual functional consequence of these variants. So just to underscore that this is a Mendelian segregation and a typical human genetics pedigree, this is an example from a few years ago, a case report for CYP2C9 and acetocumarol. Acetocumarol is a blood thinner that's frequently used in Europe, and this was a case of an individual who was very sensitive to the drug, only requiring less than 1 milligram a day. He was genotyped and sequenced for CYP2C9 and found to have two loss-of-function alleles, star 3 and star 11, who was compound heterozygous. And then you can go back and test the additional family members and appreciate how his brother inherited both normal copies from his parents and was a normal metabolizer, and both parents, of course, were the heterozygous intermediate metabolizers, where he was unfortunate enough to inherit both loss-of-function alleles. So we have to talk a little bit about CYP2D6 because it's one of the most important of the P450s, as it's involved in the metabolism of about a quarter of all medications. We get some of our testing volume from psychiatry due to its role in the metabolism of antidepressants, antipsychotics, among other agents. And thankfully, there are some practice guidelines already out there for things like codeine and the tricyclic antidepressants, as well as the SSRIs. However, it's extremely polymorphic, so it's a very difficult gene to interrogate because there are over 100 variant star alleles. It also has copy number variation associated with it, so you can have full gene deletion as well as full gene duplication. Just to show how difficult it is to interrogate, we can align the sequence of CYP2D6 with its pseudogene, which is shown here, CYP2D7. It's 97% homologous between the two. So most testing platforms require some kind of specific oligos or primers to actually pull out some of these unique regions of CYP2D6. Because of the copy number variation, it has additional categories of metabolic phenotype that we have to appreciate, and that is shown here when we infer the phenotype of CYP2D6. By using a probe drug for CYP2D6, we can look at the histogram of people's metabolic profile by using the prodrug nortriptyline in this case. And if we compare that to the genotype of these individuals, those individuals who have an increased copy number of normal functional alleles of CYP2D6 are classified as ultra-rapid metabolizers. And those in the middle are usually those who have 2 normal copies or 1 reduced function copy, which are normal metabolizers, and then the intermediates and poor metabolizers shown here on the right, who have 2 loss of function 2D6 alleles and are unable to metabolize agents via 2D6 very effectively. So with that introduction, I wanted to talk a little bit about pharmacoethnicity before getting into the implementation component, because I think it's an important thing that we all consider with respect to the patient population that we're addressing. So I'd like to start with this picture here of New York City. Some of you may have identified it due to the rectangle in the middle, which is Central Park and here in Manhattan. And what the colors represent are red dots represent 25 Caucasian individuals, blue dots represent 25 African American individuals, green dots are Asian, and yellow dots are Hispanic. And of course, we could make some socio-political demographic comments about that, but that's not the purpose today. What I really wanted to just show is that it's very important to remind ourselves that we're all very different in our backgrounds, and our patient population should be reflective of that, and we have to be considerate of the ethnicity and the racial background of our patients and what genetic variants are really more common in these different populations. The reason for that is because we know the drug response varies not just between individuals, but also between populations. And the reason behind that is largely driven by the differences in the pharmacogenetic allele frequencies between these different racial and ethnic groups. And some of the most notable examples of that we can see are the HLA alleles for the carbamazepine-induced hypersensitivity. There are some that are more prevalent among the East Asian population, and there recently been the identification of an HLA allele that is much more common among the Caucasian population. Other examples that we have studied are the warfarin-resistant mutation in the Ashkenazi Jewish population, 2C9-star8, which is much more prevalent among African Americans, and there are several variants, 2C19-star3, as well as the most recent NUG15 from June Yang's group in St. Jude's, who identified that with thiopurine toxicity that are much more prevalent in the Asian population. So these are all things that I think really underscore the importance of being really aware of who you're testing and what kind of genetic variants you're testing in this patient population. Going back to CYP2D6, we can look at the worldwide prevalence of some of these the two most kind of important phenotype categories, the ultra-rapid metabolizers and the poor metabolizers, as these are the individuals who are kind of on the extremes of that histogram, who are most susceptible to adverse drug reactions or lack of efficacy for some of the medications metabolized by 2D6. So, of course, we can't really lump everyone in North America into one population, but just roughly 10% of individuals are either ultra-rapid or poor metabolizers, which suggests 1 in 10 of the people on this call right now are one of these two 2D6 metabolic profiles. But depending on where you're from and your nationality or your ethnicity background, there are certain populations that have very high frequencies of some of these alleles in their population versus some of them that have very low frequencies, as we look up in the East Asians for both ultra-rapid and poor metabolizers. Again, underscoring the need to know your population. The last example here I wanted to show was the VCORC1 promoter variant, which is the most notable and strongest predictor of warfarin dose requirements that many of you are probably very aware of. The G to A transition allele in the promoter of the gene that confers warfarin sensitivity with the A allele. Worldwide allele frequencies are shown here, and on the left we can appreciate that the sensitive allele frequency is quite low. It's less than 10% among African Americans or those of African ancestry. But if we look at European ancestry, it's actually 50-50. It's a very common allele in the Caucasian population. But then if we move over to East Asians and the Chinese and Korean and Japanese populations, it's actually very rare to have the normal copy, as it's completely the inverse from the African ancestry populations, where the sensitive allele, the A allele, is actually the predominant allele in this population. And that is actually very consistent with what we see in warfarin dosing compared between these different racial groups. We don't have time to get into the details of the warfarin story, but I just wanted to highlight how that concept of these different allele frequencies was really driving the reasoning behind why these dosing algorithms do not perform very well in non-Caucasian populations. And for those who know the field, know the results of the COAG trial that was published in the New England Journal of Medicine in 2013, and how there was no difference between pharmacogenetic guided dosing versus clinical algorithm guided dosing. But if we look back to the details of that trial, we can kind of see why, and that's because a quarter of the population was actually self-reported black, and that the alleles that were being tested for in the algorithm were actually just the STAR2, STAR3, and that promoter polymorphism allele in VCORC1. And if we look at the frequencies of those three alleles, in the Caucasian population, 3/4 of the individuals carry at least one of these variants. But it's the complete opposite of that in the African American population. So again, going back to that clinical trial, a quarter of the population probably wasn't really being genotyped for the alleles that were most notable and most important for that population at that time. So with that, I wanted to, for the last few minutes, kind of get into clinical implementation and taking what we've learned from the background and also the concepts from the pharmacoethnicity section and try to apply that to our population here in a clinical context. So as some of you have probably done yourselves, the road from discovery to clinical implementation is not always so straightforward. And of course, within the test, we have to make sure that we have a valid test for doing our genotyping, and establishing clinical utility is a whole other discussion that I don't think we have time for today. But of course, there's a lot of regulatory oversight by the FDA, our CLIA and CAP regulations, as well as education and guidelines are needed, recommendations to do with testing results. And of course, cost and reimbursement is a very big issue, as insurers and third-party payers are not always consistent with how they are reimbursing for pharmacogenetic testing currently. So we don't have time to go through all of these issues, but I wanted to just highlight two of them. One of them was just looking at the analytical validity of test development, and the second was looking at some of the education issues and guidelines that are kind of cropping up with pharmacogenetics. And for those of you who are wanting to look for where a test is available, of course, the genetic testing registry has been live for a while now, and you can still look up pharmacogenetic tests within this public resource. So in terms of setting up your own pharmacogenetic test in your clinical laboratory, there's a nice resource available at the Coriell Institute for Cell Lines. As in 2010, Vicki Pratt published a paper in the Journal of Molecular Diagnostics deriving consensus genotype calls for 2D6, 2C19, 2C9, and VCORC1 and UGT1A1. This was a nice resource for those of us who were setting up assays at the time to have some positive controls for these variant alleles. But the problem was that there wasn't many examples that had some of the low-frequency variants that were more prevalent in other populations, as we mentioned in the last section of the talk. So since that time, we actually have completed a subsequent study to this that looked at many more different cell lines, but they were actually selected this time for the diversity in their cell lines. So they were picking up some of the variant alleles that were more prevalent among African and Asian populations. We looked at many more genes. Together, we were able to make consensus calls for some of these low-frequency variants. And again, these are available at the Coriell Cell Repository. And these will be nicely, I think, very good resources for test development and validation, as well as your QA programs, and of course, for proficiency testing. So the CAP PGX proficiency testing surveys are available, and there was a nice publication by Evelyn Wu a few years ago to talk about the results of that. I encourage you to look at them because they really underscore the variability in how labs can report some of these variants, how the testing of different variant alleles is important, and that directly can influence the predicted metabolic phenotype, and the interpretation of these results can be variable between labs. That's still an ongoing area. And speaking of reporting, it's another area I think that still needs a little bit more work, because there's not very many guidelines on how to report pharmacogenetics. So some laboratories can do their interpretation quite distinct from others. And so there are some ACMG guidelines available, as well as the ClinGen work group is also in progress to work on this, and there has recently been an international work group recommendation on testing reporting guidelines that was published in CPT earlier this year. On the education side, we've done a little bit of work here at Mount Sinai to look at our providers and their level of education and familiarity with pharmacogenetics as well as clinical decision support. And it was very informative because when we asked them questions about clinical decision support, as shown here on the left, the vast majority of them are very aware of clinical decision support, something that they use. But when we asked them about pharmacogenetics or genome-guided prescribing, they're aware of it, but when we actually ask them if they've used it, that's a much different story. So again, most of them have used clinical decision support at some time, but very few have actually used pharmacogenetics in their practice. So what that really tells us is that we have a continued need for provider education on pharmacogenetics, and also I think that there's a real need to provide them with support on what to do with these information because they really don't have the education or training to manage these kinds of pharmacogenetic data currently. So we need to give them not just the test results, but also the support and practice guidelines as to what to do with these results. So there's a very great resource for this, thankfully, which is the Clinical Pharmacogenetics Implementation Consortium. So CPIC was started several years ago by Terry Klein and Mary Relling with the aim of facilitating pharmacogenetics implementation initially through clinician education. And how they were doing that was to publish these practice guidelines. But importantly, CPIC stays out of the debate over whether you should test or whether you should not test your patients. That's a very important distinction here, and that CPIC's mantra is more so that at some point people are just going to have these data and that physicians should have some type of practice guideline as to what to do with these data when presented with them, regardless of whether if they order the test or if the patient had their own pharmacogenetic information already in hand. And so they've been working their way through a lot of these gene-drug pairs to provide really structured, really thoughtful practice guidelines that are a great resource for not just education, but also for teaching purposes as well, too, because we have structured sections on the gene, sections on the drug, sections on the evidence supporting the relationship between the gene and the drug response, and a lot of information in the supplemental material that can be very useful for those who want more information on that gene-drug pair. So at Mount Sinai, we've adopted a preemptive pharmacogenetic testing program here to deploy pharmacogenetics, and that has been driven through our Institute for Personalized Medicine and also facilitated by our biobank program, or what's called BioMe. And so BioMe is a prospective collection of DNA and plasma samples that's linked to de-identified electronic health record data. So when patients come here, they are asked if they want to participate in the biobank, and they are consented to give access to their electronic health records, as well as to provide a sample for DNA. And so in total now we have over 32,000 patients who have enrolled in this program, and they also give us permission to recontact them for further research. So this has been a really great resource for discovery, but importantly, though, too, it's really reflective of the patient population here in New York City. It's very diverse. It's basically 1/3 European ancestry, 1/3 African ancestry, and 1/3 Hispanic ancestry, basically. So not only for discovery purposes, but for implementation purposes, the biobank has been a great resource because we go back to them and we ask them if they want to participate in these pharmacogenetics implementation programs. And so there's two parallel programs that have been ongoing. One of them is called IPM PGX, and the other has been funded through the NIH as part of the eMERGE network. Some of you may be members of that as well, called eMERGE PGX. So together we have genotypes here at Mount Sinai, almost 2,000 patients. The eMERGE network in total is actually closer to around 6,000 through a combination of both just genotyping and sequencing as well, too. And then deploying some of these preemptive clinical decision support tools for a number of gene-drug pairs for these patients. So these patients are not on some of the medications already, but we collect these genetic data, put it back into the electronic health record, so when they do come back, we can provide some type of clinical decision support for those gene-drug pairs that we have selected to deploy. So in order to do this, we had to develop a fairly sophisticated information technology system, which we've called ClipMerge, which is also funded through the Institute for Personalized Medicine here, the IPM. And this is a data management system and a rules engine. It integrates genomic data into routine care. And our whole purpose initially for this pilot program was to look at provider opinions and the functionality of it. So again, just to kind of highlight the workflow, we basically collect new samples, saliva or blood from our biobank participants. And this time, instead of going to the research laboratory, it comes to our clinical Mount Sinai genetic testing laboratory, where we do the DNA extraction and the genotyping with our pharmacogenetic panel. I push these results back to the IPM ClipMerge platform, which houses a database as well as a rules engine. And that rules engine is fueled by the CPIC guidelines, essentially. And so then when these patients come back, these data are housed within our electronic health records. And if a physician orders one of these medications that we have rules for, and if the patient has an at-risk genotype, that physician at the point of care and at the time of encounter actually gets a pop-up best practice alert to tell them that their patient is either some kind of poor metabolizer or intermediate metabolizer, depending on the CPIC guidelines, and gives them options for the recommendations for alternative therapies or alternative dosing regimens. So importantly, though, we're not powered to look at clinical outcomes and things like that. We basically want to do this for the sheer purpose of implementation. And so a physician does not have to take these recommendations. It's ultimately their decision. They know better than we do. But they can't just close out of it and walk away. They have to click through and give us some reasons why they did not select the recommendations. And so those kind of data we can track very nicely. And so we're getting a lot of feedback as to how they enjoy the system, how they respond to it, what they like about it, what they don't like about it. Just to give you one example of the gene-drug pairs, this is simvastatin, and the gene that's predictive of the high myopathy risk is SLCL1B1. So if a patient comes back who has an at-risk genotype and the physician orders simvastatin, there's a whole algorithm here based on the logic to fire certain best practice alerts depending on the genotype for that individual. And then we have various smart sets that will open up and allow the physician to select the different medications should they want to, and then the actual results are filed back in EPIC as needed. This is an example of the clinical decision support that a physician would get in the case of a CC risk genotype for SLCL1B1, telling the physician to consider prescribing a lower dose of simvastatin or to choose an alternative statin based on the higher myopathy risk and to continue routine CK surveillance. So the initial program has been running now for about 2 years, and this clinical decision support system now fires probably once a week on average. And just looking specifically at the simvastatin data, I was actually surprised pleasantly so, to see how often physicians were actually accepting the pharmacogenetic recommendations. In this case, over 60% of them were actually taking these recommendations and following through with what CPIC had recommended for them. So it's very encouraging results, and we are collecting these data and hope to be publishing the updated IPM PGX program later this year. So with that, I just wanted to summarize and conclude, and then I'll pass it over to Divya. But in general, I think there's a real continued need for pharmacogenetic variant discovery among non-European populations, and this is a research comment, but it also is really based on the fact that that's going to be needed when we talk about clinical implementation, because our targeted panels should really include variants that are prevalent in the intended patient populations. In addition, ongoing provider education in pharmacogenetics is necessary to facilitate implementation. I don't think anybody would disagree with that. And the reason for that is that most providers really do not feel well equipped to manage a lot of these data in clinical care currently. And then finally, this preemptive pharmacogenetic testing model is new, but we're very excited about it, as it's the way we have chosen to implement clinical genomic medicine here at Mount Sinai, as well as other institutions. And this is really facilitated by the CPIC and other practice guidelines. But of course, it requires significant institutional investment, as well as an IT infrastructure that is not going to be easily developed, requires time to put together. So with that, I thank you for your time. I just wanted to acknowledge all my colleagues here at Mount Sinai in our department, as well as the Clinical Genetic Testing Lab, the IPM that drives a lot of the implementation work, and my colleagues here at Regeneron, who were previously Omri and Nura, previously were at the IPM, who have now recently moved on to Regeneron. Happy to take questions at the end of the talk, I believe. And Elizabeth or Allison, if you wanted to pass it over to Divya, be my guest. Thank you for your time.
Our next speaker is Divya Neelam, Manager of Applications Marketing at Agena. Welcome, Divya.
Thank you, Allison, for the introduction, and Dr. Scott for showing us the need for pharmacogenetic testing and how it's currently being implemented at the Mount Sinai through the IPM program. Thank you, everyone else, for joining our presentation this morning. And I would like to now present to you Agena Bioscience's solution for pharmacogenetic testing on the MassArray system. The MassArray system, as you know, is a proven technology, and from what our customers tell us, it is a workhorse for the lab. Our biochemistry allows you to multiplex anywhere from tens to hundreds of actionable variants, and the process of running assays is very simple with an easy-to-use assay design software where you input your markers of interest and the software designs the multiplexes for you. We can work with a variety of samples, different biomarkers, including insertions and deletions, as well as copy number variants. Our biochemistry uses simple PCR and extension reagents, and as it is based on mass differences, we do not use any fluorescence, which keeps the cost low and also makes our detection sensitivity very high. Our chip has an open layout, allowing you to set up samples and assays in a format which is most compatible with your existing lab workflow. And our platform comes in different formats with 96 and 384 chips, providing scalability for your lab's throughput needs. The MassArray system requires minimal DNA of just 5 nanograms per reaction and is proven to work with degraded and low-quality samples. We can efficiently multiplex anywhere from 25 to 30 variants in a single reaction, including insertions and deletions, and CNVs in the same reaction. So this minimizes the number of reactions and workflows required per sample. The DNA to results workflow is very simple with only 6 steps and can be easily completed in a single 8-hour shift. This 8-hour workflow has hands-on time of less than an hour. The biochemistry for the MassArray is simple, as it uses targeted PCR reactions where the amplicon length can be adjusted to be more specific. There is no amplification of the pseudogenes, and so this can come in very handy when working with genes such as CYP2D6. Also, as I mentioned, as this is a mass spectrometry-based platform, we're detecting the actual mass of the DNA fragments and bases, providing absolute detection, unlike fluorescence. And as we have all 4 nucleotides available in the extension reaction, we can detect multi-allelic variants and indels in the same reaction without any extra effort. The MassArray system comes in multiple formats to suit your lab's throughput. We have a 96 and 384 format instrument, and our newest instrument, the MassArray system with the chip prep module, automates sample conditioning, dispensing, and processing steps. So if we take an example of a typical pharmacogenetics panel analyzing anywhere from 50 to 70 variants, they are generally multiplexed within 3 wells. And with the chip prep module system, in the 96-well format, you can process almost 8 plates in a single day and put in 2 plates at the end of the day before you head home. So that allows you to process nearly 320 samples in a single day. For labs with a higher sample numbers, the 384 system can process more than 10 384 well plates within a single day. Our pharmacogenetics assays range from a comprehensive 36-gene IPLEX PGX Pro panel to gene-specific panels such as CYP2D6 or CYP2C19. If you're not seeing an off-the-shelf option that covers your needs, our experienced Assays by Agena staff can assist you in designing and implementing a custom panel. We have developed assays for more than 350 pharmacogenetic variants, including copy number variants and insertions and deletions. And also, for the maximum level of flexibility, you can utilize our online assay design suite tool. Just insert the RS numbers for your targets of interest, run them through the software to get a multiplex panel designed within the hour. And all you have to do is then order oligos from a vendor of your choice, use our reagents, and you're ready to start testing in the lab. Our pre-designed panels are used worldwide. The iPlex PGX Pro panel offers maximum coverage with over 200 assays across 36 genes, and this is multiplexed into 8 wells. This panel covers over 99% of the pharma ADME core list of variants. If you're looking for something more targeted and actionable list of variants, the PGX 68 panel covers the major haplotypes from 16 genes, including 5 copy number assays for CYP2D6, and this panel is multiplexed into just 3 wells. Here are the haplotypes covered with the iPlex PGX Pro panel. As you can see, this is a very broad panel and would be a great starting point to first screen patients for your studies. This is the coverage from our PGX 68 panel. As you can see, it is fewer number of genes, but we still cover the major haplotypes within these genes, providing a very actionable list of variants. In addition to our simple and streamlined workflow, data analysis is also very straightforward with the mass array system. We provide simple, easy-to-use reports which can be generated on your PC with any of our pre-designed panels, as well as for your custom panels that you would like. The report provides genotypes or diplotypes, as well as haplotypes for each sample, and also copy numbers. In addition to text-based reports, we also have graphs and spectrum-based analysis for easy troubleshooting. Through these graphs, you can easily differentiate samples with varying copy numbers, and the software also provides probabilities for each call. To summarize, the ability to multiplex 25 to 30 variants or loci within the same reaction, together with copy number variants, minimizes the number of reactions required per sample. With our database of robust and validated assays, there is no need to test samples in duplicates or triplicates. These factors together greatly reduce your operational costs and also make your technician and lab manager very happy. And what's really nice about the mass array system is its flexibility. As the chip content is not fixed, you can run other tests on the same chip, such as carrier screening for cystic fibrosis or susceptibility for heart disease. The mass array system is a proven technology with over 2,500 publications, and here are just a snippet of some of our recent publications in pharmacogenetics. As you can see, they cover pharmacogenetics from drug response, resistance, toxicity, and pharmacokinetics.